and we'll look at different uh, things here that I don't have up because it's hard to have these, so we're going to have to show this in class. Okay, and we're skipping on to the next part. The next part is, how does this molecule actually get used, not to copy itself, that's what we just looked at, but to make proteins that cause traits, the second function of this molecule, to be like a recipe, okay? Well, it was figured out that almost by common sense that we have a location problem. This DNA in eukaryotic cells stays in the nucleus, but the little creatures that make proteins, the little organelles called ribosomes, are outside the nucleus. So if this is a recipe to make proteins, and these are the things that make proteins, how does it work? The DNA recipes never come out here. Well, it had been discovered, a good while back, that this other nucleic acid, RNA, was found both in the nucleus and out. So it correctly and logically was figured at first to be, this must be the thing that takes this recipe out to these ribosomes, which are now going to be like the chef in our analogy, right? It must be this other nucleic acid. It has a sequence of bases. It also, therefore, can act as a recipe and information, right? Somehow it seems that DNA is used to make RNA. That's the process we're going to call transcription. And then that RNA travels outside and is used by ribosomes to make proteins. Being used by ribosomes to make proteins is called translation. So DNA gets transcribed into RNA, RNA gets translated into proteins. And in both of those steps, it's the sequence of bases that makes a difference. So here we now have to look at RNA real fast, right? RNA has a couple differences compared to DNA. One is that most of the time, RNA is single-stranded and DNA is double-stranded. So RNA is just one string of nucleotides. RNA has one different base. RNA doesn't have thymine. RNA has uracil. Like thymine, uracil connects only to adenine by 2 hydrogen bonds. I should have dotted them, make them hydrogen, right? So uracil is complementary to adenine. And then the other difference is in their names. R is for ribose, D is for deoxyribose. They have a slightly different sugar. That makes a big difference, but a slightly different sugar. The key workings of these molecules, both DNA and RNA, again, are all about the bases and the sequence of bases. So that's what we focus on the most. Okay, so now, here we have DNA acting as a template now to make RNA, not more DNA. That process, again, we call transcription. Is this RNA going to be identical to the DNA that was used to make it? No, right? It's got a different sugar for one thing, but if this is the DNA, let's say this small strand is CA, I'm going to be simple here, CAG. And I use this side of a DNA molecule to make this RNA, the sequence of RNA bases would be the complementary ones. G, if this is A, remember U is going to be complementary to that. C. So no, this RNA is not identical to this DNA. It's complementary to it. That's what you would call them. The DNA is used as a template to make complementary RNA in the process called transcription, which also occurs in the nucleus, right? Because that's where DNA is. Any process with DNA involved occurs in the nucleus of a eukaryotic cell, okay? So we're going to see that it turns out RNA has several different kinds of RNA that have several different functions. We're only going to focus on 3 in this class, But the one we're going to focus on the most is that one that's going to be used as the recipe by the ribosome. And that's called messenger RNA. Early on they called it that because that's the one that takes this message out of the nucleus to where the ribosomes are and gets used as like the recipe message. Okay, so what's going to happen in translation with this ribosome? This ribosome is going to take this messenger RNA and its sequence of bases, and it's going to use them to put together a sequence of amino acids, right? Very simple. Here you have two very similar words, transcription, translation. You have to keep those trans words straight, which is which. So now, this idea that we've said, now we're going to nail it down, is called the central dogma of biology. Dogma is a fundamental understanding, belief, concept, whatever you want to call it, often used in religious talk, right? That is fundamental to the understanding of something, in this case, biology. The central dogma of biology is this: DNA is used as a pattern to make RNA. Now notice, I used an arrow here, and that's commonly kind of used, but this is not one of those chemical reaction arrows. DNA does not turn into RNA. That would be a misconception idea. DNA is used as a pattern to make RNA. The sequence in its bases is used to put together a molecule with a complementary sequence of bases. RNA then, and its sequence of bases, is used to make a protein, a certain sequence of amino acids. Again, it's all about sequence. And then proteins are what cause traits. This is how we finally figured out, we humans, how you get green eyes. You got the DNA from your mom and dad, right? You used it to make RNA, used that to make protein, had the protein that caused that reflected light to make your eyes look green, or whatever they are, right? So now go back to this. What does this arrow then symbolize with process? Transcription, right? What does this arrow symbolize? Translation, right? And translation is also known then as protein synthesis. We will call them both that pretty much interchangeably. Not pretty much, they're exact synonyms. We'll use them pretty much half the time. Okay, so there's the central dogma. In another diagram that they like these for EOC tests, right? You have the names of the processes here. Again, you have an arrow that is not a chemistry reaction arrow, although these are chemical processes. DNA is not the thing, excuse me. DNA is not the reactant that turns into the product RNA. So this arrow doesn't mean that, just so we're sure, right? Here we're going to have a concept that we're coming up with soon after we look at transcription a little more closely, real fast. Here we have a process that has three bases form kind of like a word that says put this one amino acid here. It turns out this language ended up being a language of three-letter words, so to speak. Little sections of RNA, 3 bases long, 3 nucleotides long with 3 bases. We're focusing on the bases, right? That say put a certain amino acid here. These are all shortened abbreviations for amino acid names. Okay, so that's the central dogma in diagram style. So here's a more close-up picture of what's happening inside this RNA polymerase in its active site, right? Its active site is just the right shape to hold a template strand of DNA. This would be the template strand, right? Remember, only one side is used for this transcription. And here's the nucleotides, which are everywhere in here. There's nucleotides all around. They're moving around at random, right? But it's not going to take very long for an A nucleotide to bump into the right spot where it's going to bond with a T, hydrogen bond, and then it's going to bond the sugar phosphate part with this sugar phosphate part. That's what really connects these nucleotides to each other in a strand. This strand is then going to disconnect from the DNA, and eventually when this process is all done, you'll have this separate piece of RNA, and your DNA will zip back up. This makes this process very much like recipe stuff, if I use this analogy, right? So if I have one of you in class, and let's say you have in your kitchen in your house, you have a recipe book that's in Spanish. Well, it's got a chocolate cake recipe in there that I want to make, but I'm out here. This is where I am. I'm out here, and you won't let this recipe book out of your house. This recipe book is like the DNA, right? It's got this one recipe I want in it, right? But you won't let the recipe book out of your house, out of the nucleus in this case, right? But if you will be so kind as to open your recipe book to one place and use that Spanish language to make me an English language recipe, this would be the RNA, specifically the messenger RNA, and then send that in the mail to me. I'm a ribosome out here, right? Then we can make this work, right? So that's what this process is like. You're going to transcribe this from DNA language to RNA, from Spanish to English. Because if you sent me that Spanish recipe, I wouldn't be able to use it anyway. I need the different language, complementary language message. This is the process that's evolved to give you your blue eyes, green eyes, brown eyes, whatever you got.